In this video, I show you a method to distinguish islets from exocrine tissues. We stain islets with Dithizone when counting islets. Here, red particles are islets stained with Dithizone, and the remaining yellowish particles are non-islets, or exocrine tissues. Dithizone staining are toxic to all cells including islets. Therefore, basic researchers using human islets conduct hand-picking islets under microscope without Dithizone staining. However, it is not easy. My microscope magnification is set at 2.5. The image is captured to my computer screen. Here, you can see on this computer screen is a standard image of islet preparation. Now, I have a magic black sheet. On the sheet, I pour some special solution. Then, I put my dish slowly on it. I swing my dish to move tissues in center. Now, under this condition, I acquire the image by clicking this tab. Here you go. This is standard method. This is my modified method. White particles are islets. Again, standard method. And my modified method. This is Dithizone staining from the same preparation. What did I use? A magical black sheet is from this binder, and my secret solution is just water. Thank you for watching.